In the previous chapter, we considered the process of using both strands of the DNA double helix as templates to synthesize two new daughter strands of DNA. In this chapter, we will look at the process of using one strand of the DNA as a template to synthesize an RNA molecule. Transcription is the first process in the expression of genes, most often completed in the translation of the RNA message into a protein. The enzymes that catalyze transcription are RNA polymerases, and they use one of the DNA strands as a template to synthesize the RNA complement. Since an RNA polymer is generated, the substrates are ribonucleotide triphosphates. As in the case of DNA polymerases, and for similar reasons, the magnesium cofactor is required. Another similarity to DNA polymerases is that RNA polymerases read the template in the 3' to 5' direction and synthesize the complement in the 5' to 3' direction. A dissimilarity, however, is that RNA polymerases are capable of de novo synthesis. They do not require a primer. If we examine the core enzyme in E. coli, pictured below, we find that it is a multi-subunit complex, composed of subunits alpha, beta, beta prime, omega, and sigma. The core enzyme is responsible for the catalytic function and lacks the sigma factor. The combination of the core enzyme with the sigma factor constitutes the holo enzyme. It has all of the component parts. There are two DNA strands, but only one is used as the template, and it is appropriately known as the template strand. It is also denoted as the antisense or negative strand. The RNA molecule produced by reading this template is an RNA copy of the sequence in the opposite DNA strand. Since the sequence in the RNA contains the code that will later be translated into protein, this DNA strand is known as the coding strand, or the sense or plus strand. Notice in the figure below that the top DNA strand is the coding strand and that its sequence matches that of the RNA transcript, with U's in places of T's. The holoenzyme is responsible for binding to a specific region on the DNA in order to initiate transcription. This region is known as the promoter and is specifically recognized by the sigma subunit. After the initiation of transcription, the job of the sigma factor is accomplished and it dissociates from the RNA polymerase complex. In our next lesson, we'll look more particularly at these promoter regions.